All right, so we had protein synthesis, and we decided to make models of the flow of genetic information first out of these cookies, which represent cells. So first, we find the DNA inside the nucleus, and then it goes through the process of transcription, which is explained here, which we'll explain later, and it creates mRNA, which then leaves the nucleus and goes through the process of translation and creates amino acids, or proteins, out of amino acids. All right, so.
All right, so when tRNAs bind to the mRNA, they bind by matching the complementary base pairs on anticodons on codons. This is the chart of the codons, and each specific codons code for specific amino acids.
All right. Okay, so we will start with transcription, and this cake is made out of is a pound cake. All right, so we'll start. RNA polymerase attaches to DNA, which was unzipped by RNA polymerase. And then it attaches to the promoter sequence here. And so as the RNA polymerase is moving down the promoter sequence, the matching nucleotides come in the, like, for example, oh, never mind. So the matching nucleotides would come in, and so you can see here and produce a strand of mRNA, and then so the nitrogenous base pairs are A and U, and then T matches with A in DNA, and C matches with G.
And then G matches with C.
And G matches with C. Okay, and then the RNA, then after it goes through this, then leaves the nucleus through the nuclear pores, and the DNA reattaches. Right. And then after it leaves the nucleus.
And then it goes to the cytoplasm, where it binds to the ribosome at the start codon, which is AUG, and then tRNA, as shown here, tRNA finds the complementary finds the base pairs on the mRNA and matches its anticodon with the codon as by the chart shown here. And what happens is there's amino acid at the end of the tRNA, so after it binds to the mRNA, it shifts down the mRNA strand, and another tRNA binds to the mRNA. And the amino acids at the end of the tRNA form a polypeptide bond, and after it does that, the first tRNA that came onto it leaves. Leaves, and then leaves into the cytoplasm to find another amino acid specific to that tRNA. And what happens is this process repeats until, as shown here, you have a whole long chain of amino acids.
Right here. And as you can see, we have the pink one, which is representing methionine, which is always the start, which is the starting amino acid for AUG, which is the start codon. And this goes around, this shows the process when the RNA leaves the nucleus, and it goes around, and it keeps binding to new tRNA keeps binding to the mRNA and producing amino acids. And finally, when it reaches a stop codon.
Mm-hmm. And then after it reaches there, no amino acid is added to the chain, and everything disassembles, and
so the ribosomal subunits disassemble, and the tRNA disassembles,
and the amino acid folds into the protein, which can be used elsewhere.